Hello, welcome to a quick overview of protein synthesis. Proteins are made from amino acids, which are made from RNA, but the RNA is also made from DNA, and we call this process as central dogma. Now, in the eukaryotic cell, we know that the DNA is inside the nucleus. And if you look at this picture, we have the DNA here. A short part of the DNA, this area we call a gene. And to start with, an enzyme called RNA polymerase is going to attach to the gene. And it is going to copy one part of the gene by adding RNA nucleotides. Finally, it's going to make a copy which we call the messenger RNA or mRNA. Now we can see that the gene has two strands, the top strand and the bottom strand, and we use only one strand to make the messenger RNA. We call the strand which we use to make RNA or messenger RNA as the template strand, and the one which we did not use as the coding strand. Now this process is called transcription when we copy the DNA into messenger RNA. After this, the messenger RNA will go into the cytoplasm. Before we look at what happens in the cytoplasm, let's take a look at the messenger RNA. It has different RNA nucleotides, as you can see here. And when we take 3 of them, we call that a codon. And we can use this chart to help us to match a codon to an amino acid. One codon will match to one amino acid. Example, the codon AUG, you can find here AUG equals the amino acid methionine. And ACG equals threonine. We have GAG will be glutamic acid, and then we can look for the other amino acid. Finally, this AUG, this codon, is called the stop codon because it doesn't have any amino acid. So let's look at what happens to the messenger RNA when it gets to the cytoplasm. When it goes into the cytoplasm, it's going to join to a ribosome. Now, a ribosome is a type of RNA which we call ribosomal RNA or rRNA. It has a small subunit and a big subunit. The messenger RNA will go between the big and the small subunits. Inside the cytoplasm, we have another type of RNA called the transfer RNA or tRNA. This is a picture of a transfer RNA. Now, transfer RNAs carry amino acids, and different transfer RNAs carry different amino acids. At the bottom of their structure, we have three RNA bases which we call anticodon. And the transfer RNA will bring the amino acid into the ribosome, and the codon of the messenger RNA is going to match with the anticodon of the transfer RNA. Now the ribosome will help the amino acids to join together to form a long chain of polypeptide. Now, when we have a long chain of this polypeptide, it's going to fold and then form the protein. We call this process as translation, when we go from messenger RNA to create a protein. Thank you very much, and you can start your activity now.